presentation on my mobility experience that I'm actually currently undergoing. So a little bit of background before I get into the mobility. So my research involves investigating the interaction of corrosion byproducts with human blood proteins, specifically the protein albumin. And this can be metal ions or metal nanoparticles. And the reason this is important is because metals, even those that are conventionally considered inert or safe, may have some fairly devastating effects on human health. So this can include things like inflammatory responses, like pseudotumors or the development of metal allergies. Metals have been implicated in neurodegenerative diseases like ALS, Alzheimer's, and Parkinson's, and metals have been implicated, or metal exposure has been implicated in the development of cancer. So all around, it's a fairly important area of investigation. So with that really brief background out of the way, let's talk about where I am and what I'm doing. So I am currently in Colorado State University, which is in Fort Collins, Colorado. I'm in Debbie Kranz's research group where they study vanadium, and specifically vanadium chemistry in biological systems. And the reason I'm studying this is because vanadium is present in the most common form of titanium biomaterials, the Ti6-4 alloy, which is 4% by weight vanadium. So this biomaterial is incredibly prevalent and sees use in many areas of the biomedical device field. And that can be everything from pacemakers, arthroplasty implants like knee or hip replacements, spinal fusion cages, and a lot more. Now, the concerning part is that it's been found that within as little as a day, vanadium is completely depleted from the surface oxide of this material, suggesting that it's leaching into the human body, and the exposure to this metal may be a lot more severe than you might suspect. So vanadium is known to bind to the protein albumin. In fact, there's already been a lot of research into this area because vanadium has a use case as a diabetes drug. But in that situation, vanadium has ligands attached to it. It's a metal ligand complex drug. And I'm more so interested in the free metal. Unfortunately, as soon as I started this project, I kind of ran headfirst into a wall. So it turns out vanadium speciation is complicated. It's really complicated. So this Poiret diagram I put up there is really just the tip of the iceberg. The human body is a reducing environment. So if we take a fairly extreme reduction potential for a physiological environment, glutathione's reduction potential, which is negative 240 millivolts, and we model the vanadium speciation, the only species present is vanadium in the +4 oxidation state. Specifically, we see this dihydroxy vanadyl species. At the reduction potential of whole blood, which is around negative 90 millivolts, we see the same species present. If we try and simulate physiological conditions ex vivo, you'll have a solution that has no reduction potential. And in that case, vanadium will take the form of a polyoxovanadate in the +5 oxidation state. Polyoxovanadates are basically just clusters of these vanadate ions together, and that's what's pictured in the slide there. Now, if I add reducing agents, I will get vanadium-4, but what's really important here is I will not get the dihydroxy vanadyl species. I get some other different complexes that are not physiologically relevant. Now, to make matters worse, in the human body, vanadium undergoes something called vanadium recycling. This is where a bit of vanadium-4 will come in contact with a biological oxidant like a reactive oxygen species, reactive nitrogen species, or some other biological oxidant like, I don't know, vitamins in some cases. The vanadium will be instantly oxidized to vanadium-5. And then because it's in a reducing environment, it will be nearly instantly reduced back down to vanadium-4. So it has this really incredibly complicated speciation in physiological conditions. And that makes trying to simulate it a real nightmare. And that's essentially why we've moved on from this project. So I'm going to try and do something else while I'm here. So my new focus while I'm here is to determine the exact species of vanadium that gets released from corroding Ti6-4 alloy material. Now, there are some very interesting vanadium-specific techniques. V51 NMR is really quite a fascinating one, but unfortunately, it won't work for this since the concentrations of metal released is far too low to detect. But EPR, or electron paramagnetic resonance spectroscopy, should provide us with some interesting and quite novel data. So that is what I'm going to be doing while I'm here. Thank you very much.